Hello, everyone. I am Kenny Coogan, the Education Director for the International Carnivorous Plant Society, and we have a really good turnout today, so I'm very excited. Thank you, everyone, for joining us live. And if you are live, you can type into the chat box questions for Jana. And I met Dr. Jana Libantova right after the ICPS conference in Vienna, Austria, and then I snuck over to Slovakia, and she was a wonderful tour guide of her facility, and we had wonderful conversation over tea. And now I'm excited that everyone gets to experience her knowledge. And welcome, Jana, and we're looking forward to your presentation.
Thank you for the word, and thank you for the invitation. So I work at the Plant Science and Biodiversity Center, and I pay attention to the research of Sundew for more than a decade. And we pay attention mainly to chitinases, which we belong the attention at this presentation. Firstly, I would like to present very shortly the institute where our experiments are conducted. Institute of Plant Genetics and Biotechnology was established in 1990 in Nitra in Slovakia, and since 2017 has been part of the Plant Science and Biodiversity Center of Slovak Academy of Sciences. The main mission of our institute is to advance research on plant genetics, plant growth and development, as well as plant biotechnology. Our institute has two departments. I work at the Department of Molecular Biology and Biotechnology, where three working groups operate. The group of carnivorous plants pay attention to the hydrolysis involved in the insect prey digestion. The second group is the group of parasitic plants. They pay attention mainly to the investigation of the proteome of germinating seeds of selected parasitic plants. And third group of conifers, pay attention to the investigation of the role of the extracellular proteins in early stages of somatic embryogenesis of Pinus nigra. Some words about me. I have obtained a master's degree in fermentation in bioengineering at Slovak University of Technology in Bratislava, and my PhD degree in molecular biology at Slovak Academy of Sciences. My research is focused mainly on plant chitinases and beta-1/3 glucanases, and elucidation their role in plant defense, embryo development, and digestion of carnivorous plants. Secondly, we also are searching the candidate genes of these hydrolysis in application in biotechnology. In our research, we use the tools like molecular biology techniques, protein engineering, biochemical methods, and transgenic plants. Concerning the carnivorous plants, we pay attention on sundews. Sundew grow in nutrient-poor soils, therefore they develop the system involving the glandular leaves, using which they are able to attract, capture, and digest a caught insect. In our experiments, we use the plants that are aseptically grown in vitro condition. For this purpose, we cultivate these plants on MS medium supplemented with vitamins and sucrose. For this purpose, we cultivate the plants Drosera rotundifolia and Drosera binata. Concerning the carnivory in Drosera plants, we pay attention to the investigation of enzymes involved in the carnivory. with the focus on chitinases. We want to elucidate how chitinases production is stimulated during the digestion. How do digestion products affect the gene expression of chitinases? What are the specific characteristics of these enzymes? How these enzymes from carnivorous plants can be used in biotechnology. Chitinases are hydrolytic enzymes that digest chitin, long-chain polymer of glucose, and a derivative of glucose. The chitin is component of exoskeleton of many arthropods, insects, and fungi. In our experiments, we pay attention mainly to the endochitinases. Chitinolytic enzymes involve several several enzymes like glucose amidases, phytobiosidases, and endochitinases based on the products they release from the chitin chain. Glucose amidases, they produce monomers, phytobiosidases, they produce dimers, and endochitinases, they produce phytooligomers of different lengths. Firstly, we have investigated the chitinolytic enzymes in Sundew plants. We have found out that the Sundew contains different chitinolytic enzymes in individual organs. Endochitinases and phytobiosidases contribute to the overall chitinolytic activity in plant tissue extract. And you can see that mainly in the roots and flowers that there is obvious activity of phytobiosidases. But plant physiology of these enzymes, the role in plant physiology of these enzymes still remains a bit elucidated. Endochitinases, glucose amidases, and phytobiosidases were detected in leaves, but they were not detected in digestive fluid of the Sundew plants, unlike of endochitinases. Endochitinases are present in the participate in the digestive processes, but the other chitinolytic enzymes, their role was not confirmed so far. In addition to endochitinases, there are also the other enzymes that are secreted by tentacles and playing a role in digestion of macromolecules present in insect prey. There belong esterases, phosphatases, proteases, nucleases, and other enzymes, and they digest the macromolecules into the simple molecules. The enzymes that are involved in the hydrolysis of the insect prey can have the activity or expression only during the digestive processes, and this expression is regulated even during the process of the digestion. When the nutrients are obtained from the caught insect prey, these molecules are absorbed through leaf cuticle by specialized transport proteins. In 2005, it was not clear if the chitinases involved in the digestion of the insect prey are of plant or microbial origin. For this purpose, we have placed the leaf blades onto the medium containing chitin as a substrate. 48 hours after application, we could observe the enzyme activity as a result of released enzyme from the tentacles. The presence of chitinases of plant origin was also confirmed by in situ hybridization. For this, we have used chitinase straw mRNA probe, and we have detected the chitinase signal in the sessile glands. Here you can see the presence of chitinase in non-induced plant, where the chitinase signal was present mainly in the central tracheids. When the plants were induced by mimicking the presence of insect prey, the signal for chitinases was detected in secretory cells. This was the evidence that the chitinases of plant origins are directly involved in the processes of insect prey digestion. Later, we have isolated the genes for chitinases with the role in the processes of insect prey digestion. We have found out that the expression of these genes is focused mainly to the leaf blades. In other organs, the expression of these genes is almost completely suppressed. We have obtained the same expression profile for the chitinase involved playing role in digestive processes and characterized in Drosera rotundifolia as well as in Drosera binata. Next, we have analyzed the expression profile of these chitinases when on the leaves were placed different compounds. You can see here you can see the expression in control non-induced plants. When, for example, cellulose was placed on the leaf blades, the expression of chitinase was unchanged. But, for example, sand that result that causes mechanical irritation, or laminarin producing the glucose very easily can result in suppression of chitinase in the leaf blades. There were also the other compounds. In contrast, they caused or they resulted in increase of expression of chitinase involved in the insect prey digestion. Here are specific substrate for chitinases chitin, but also non-specific compounds like pacinan. This is the polymer of glucose, or gelatin. That is substrate for proteases. All these compounds placed on the leaves resulted in expression of chitinase. So this result indicated that when we place on the leaves the potential substrate, there is many hydrolytic enzymes that are overexpressed in coordinative way. Next, we have analyzed time course of chitinase expression upon application of pacinan. And you can see that 24 hours upon application of substrate, the expression of chitinase culminated and then dropped down. This result showed that the expression is dropped down probably due to presence of the product of hydrolysis. This hypothesis was also confirmed when we have placed glucose or glucanac on the leaves, and we have observed these are the product. Both these compounds are product of hydrolysis, and both these compounds resulted in decrease of expression of chitinase under the basal level. Finally, we have placed on the leaves the product of hydrolysis and also polymeric substrate. And, expression of the chitinase was suppressed, probably due to presence of nutrient. So this result showed that when the nutrients are present in the leaves, the absorption is prioritized before further digestion of the potential substrate. A second part of our experiments was devoted to the characterization of the chitinases participating in digestive processes. For this, we have to isolate purified protein in sufficient amount. For this purpose, we have cloned cDNA form of these genes into the bacterial expression vector. This vector was introduced into the bacterial expression strain, and upon induction, we have obtained overexpressed recombinant protein of Drosera rotundifolia chitinase and Drosera binata chitinase. These recombinant proteins were subsequently purified and used for the biochemical characterization. It's necessary to say that both these enzymes have very similar enzyme structure, protein structure. Both contain glycohydrodomain that is responsible for the cleaving of the substrate, and chitin binding domain that is responsible for the trapping the substrate. In addition, both these chitinases are extracellular. In plants, there is many classes, 7 classes of chitinases, but this class of chitinases was typical for the participation in insect prey digestion. Both these enzymes have, as I have told, both these enzymes have the same structure, and amino acid similarity was more than 80%. Both these enzymes have some common characteristics and some different. Both these hydrolyses were stable, and because their half-life time at 25 degrees was more than 24 hours. Both these enzymes are very stable under 40 degrees. So you can see that Drosera rotundifolia chitinase has a temperature optimum 38 degrees and Drosera binata approximately 25 degrees. But both these enzymes, the decrease of the activity when the temperature is under the 40 degrees is very mild. On the other side, we have found out that these enzymes with very similar structure had different pH optimum. Drosera rotundifolia has the maximum activity at pH 6. Drosera binata had the maximum activity at pH 3. Both these enzymes differ also in their substrate specificity. Drosera rotundifolia chitinase was able to digest chitin in its powder form, and also it was able to digest chitosan. Drosera binata chitinase was only to digest the chitin in its swollen form, acidic treated chitin. This result indicated that in the digestive fluid of carnivorous plants, there can be different endokitinases with different characteristics that are able to cleave different type of substrate under different conditions. Very interesting knowledge we have obtained when we have characterized the enzymatic enzyme activity of chitinase, and chitinase then was fused to thioredoxin. Thioredoxin is small molecule that is present in many expression vectors, and this protein can help to stabilize recombinant protein in bacteria. But in case of chitinases, fusion to the thioredoxin increase double the enzyme activity. Catalytic efficiency of the fused form of chitinase was even triple. Then we analyze the protein by 3D structure. We have found out that tyrosine present in chitin binding domain had a different position stereometry. So that means that probably such stereometry improve the affinity of the enzyme to bind the substrate. We can also see that Drosera rotundifolia chitinase fused with thioredoxin had a better catalytic efficiency than Streptomyces griseus bacterial chitinase that is considered as a very strong bacterial enzyme able to digest the chitin substrate. Next, we have analyzed also Drosera rotundifolia products of hydrolysis, chitinase product of hydrolysis, and we have found out that hydrolysis of chitin leads to the production of the chitoligomers with polymerization of degree from 3 to 6. This is the chitoligomers with low polymerization degree can be used in the wound healing, drug delivery, or anti-cancer therapy. This type of the experiments we plan to do in the future. But at present, we have analyzed the potential of Drosera rotundifolia chitinase to inhibit the growth of specific fungi. You can see for this we have performed quantitative in vitro assay, and you can see that that purified enzyme was able to inhibit the growth Fusarium, Trichoderma, and Alternaria, but not Rhizoctonia solani. So this result showed that the chitinase from Drosera rotundifolia has the capability to inhibit the growth of selective phytopathogens and can be used in plant defense fortification. The measure to inhibit the growth of selective phytopathogens depends on the availability of the naked chitin in cell wall of fungi and the percentage of the chitin present there. Finally, I would like to sum up our results. Our research showed that chitinases with very strong enzymatic activity play a crucial role in decomposition of insect prey in sundews. The expression of these chitinases is upregulated in the presence of substrate and downregulated by the product of hydrolysis. All these chitinases, both these chitinases, we have isolated and biochemically characterized, and we can sum up that they have a very these enzymes are very stable, showing half-life times more than 24 hours. We have also found out that analyzed or characterized chitinases from Drosera rotundifolia innata exhibited different pH optima and substrate specificities. We have also found out that Thioredoxin can increase the activity of enzyme activity of recombinant chitinase. Finally, we have found out that chitinase from Drosera rotundifolia can find a potential application in biotechnology because we have demonstrated the capability to inhibit the growth of certain phytopathogens. Finally, I would like to show you the members of our current working group. Also, there are also the members of the groups of our working group that participated in our research in the past, and lots of, for example, the analysis of the product of hydrolysis we have performed with, in collaboration with a colleague from other working group, and in situ hybridization we have performed this using help of our colleague from other department. Our research was funded from Slovak and international project agencies. And that's all I wanted to present, and thank you for your attention.
Thank you, Jana. That was wonderful, and I'm so happy members of your team were able to join us, and lots of Europeans were able to join us this morning in the United States, but this afternoon in Europe. All right, so I'm going to briefly talk about the ICPS, and for everyone who is attending live, if you have questions for Jana, please type them into the chat box and I will read them to her shortly. So I met Jana just a month or two ago after the 2024 conference. We are going to be having our next conference in the year 2026. It's going to be in the Americas. If you have an organization or a university or a carnivorous plant club in North, Central, or South America, and you would like to host the conference, the deadline is approaching, and you can email Brent to learn more. The ICPS is a group of over 1,200 paid members. We're international, and we like to do lots of activities. One of them is to host monthly webinars. Our next webinar is going to be in September, and it's going to be by Dr. Nick Tippery about the genetic identification and diversity of Utricularia in North America. If you're interested in presenting, you could be a nursery owner, you could be a grower, or you could be a scientist. You can send me an email, and we would love to host you. If you go to our website, carnivorousplants.org, you will see that we have 5 animated videos. They are for the people who are getting into the carnivorous plant hobby. They're also for kids. And we also have worksheets, activities, word searches that are associated with these videos that you're welcome to download and use for free. The first Wednesday of May every year is World Carnivorous Plant Day. On that day, we post over 20 videos from scientists and growers from all over the world. You can go to our YouTube channel to check out all of those videos. Because we're going on to the 5th year, we have probably more than 100 videos about carnivorous plants on our YouTube channel. To help us keep going, you can become a member of the ICPS. You can be an e-member or you could be a regular member where you get a quarterly newsletter, full color, shipped to you worldwide. It's very high quality. Also, if you become a member, you can access the last 30 years of our Carnivorous Plant newsletter, which has a lot of great information. If you're a scientist, you are also welcome to publish your scientific findings in our newsletter. A member benefit is that you can go to our seed bank and you can get seeds shipped to you worldwide. Our seed bank is only as good as what our members make it. So if you have surplus seeds, you can send them to our seed bank manager and they will be able to disperse them. All right, Jana was able to bring a lot of Europeans to this webinar, which I'm very excited about because this month, August, we have the Carnivores in the Classroom grant, where we fund about 50 teachers to add carnivorous plants to their classroom. So this is for teachers who teach kindergarten through 12th grade. And you can learn more about it by going to the link below. But historically, we've had a lot of United States people apply and win, but we want to fund teachers all over the world. So if you're in a European country, South America, Africa, Australia, you can apply. We want everyone to have carnivorous plants in their classroom. The way that a teacher would be funded is they submit a lesson, and then we will provide $150 worth of carnivorous plants, or a carnivorous plant nursery will ship you $150 worth of carnivorous plants right to your classroom. If you would like to support this project, you can go to the link below and you can donate money. And these are the nurseries that are funding most of our teachers this year. Some of them are from the United States. Then we have European nurseries, and then we also have an individual in Australia. You can also support our education and our conservation initiatives by going to our online store down here, which ships worldwide t-shirts, mugs, tote bags, and all of that helps our education and conservation initiatives. And with that, thank you for my little ad. And Jana, David says thank you. And do you have any timelines about when you would be applying your research to the drug therapy and the cancer applications? Or is that a different department?
So it's difficult to say because at present we are only in the first stage of our experiments. We have characterized, and in the future it's possible we will pay attention in collaboration with other specialists in characterization of anti-cancer activity of these enzymes, of the products of hydrolysis or chitinases itself. But it is the research we will be performed maybe in the future. So.
And did when you started this research, why did you start looking into the sundews?
Why? Because these plants we were able to cultivate in vitro conditions. It was the main reason, because we wanted in first experiments to confirm if the chitinases are of the plant origin or the microbial origin that are involved in the processes of the insect digestion. It was in 2005. And for this, we needed the plants that were cultivated in vitro to omit the presence of microbial chitinases. So this was the reason, and from 2005 we continued with this subject of the research until now.
Jana, can you see the chat box? Okay, so I'm going to read it for people who are watching it on YouTube. But Victoria would like to know, you mentioned that the presence of product plus substrate might lower the chitinase expression because of the high nutrient. But do you know how plants might control that? Some inhibiting or signaling pathway? So I think they're asking, you know, they make chitinase to break down their food, but when they have high nutrients in the substrate, they're not producing as much chitinase. And do we know why plants or why sundews do that? Just Sarah?
That's interesting question. The presence of the product of hydrolysis decrease the expression of chitinase participating in digestive processes. But what is the signal? Maybe the product of hydrolysis that are absorbed by the plants and increase glucose level can be the signal for the controlling the expression of chitinases on other hydrolysis involved in the digestion processes. So this is such a very simple explanation. I don't know detail how about these processes, but we have only observed this phenomenon and exact explanation. It was maybe more research.
Okay, Jana, now we have lots of good questions coming in. As soon as the first question comes in, you get more. Maxim would like to know, how do you think of chitinase expression patterns, which you discovered, tissue specificity, substrate, product induction, are they unique to sundews or perhaps something similar is happening in other carnivorous plants like pitcher plants? Because we do know that lots of carnivorous plants have chitinase. So the question is, how do you think the chitinase expression patterns, are they unique to sundews, or do you think all well, the problem is carnivorous plants evolved 8 or 9 different times, so do you think the chitinase expression pattern is unique?
I think that the chitinase profile expression for the chitinases involved in the digestion can be also, that we have observed in sundews, can be also observed in other species, carnivorous species, because these chitinases that participate in the digestion processes evolve from the chitinases participating in defense processes. But these enzymes have a very high and strong enzyme activity, so their presence and expressions in individual organs must be regulated. And these enzymes can, with a strong enzyme activity, can be also in other plant species, carnivorous species, used in the processes of insect prey digestion. So I suppose that there will be the chitinases also in other plant species with the role in the insect prey digestion that will have very similar expression profile.
And non-carnivorous plants also have chitinase to, you know, fend off fungi.
Yeah, there were described the chitinases. They play a role in defense processes of the plants, but these chitinases have a the enzyme, this very high active enzyme activity are used in plant defense in different way. So that means that chitinases of class 1 that play a role also in plant defense processes, they have vacuolar localization. And the chitinases in carnivorous plants that are used, they have the same structure and very high enzyme activity in carnivorous plants are used for the processes of carnivory. That means that, for example, targeting of the chitinases is strong activity when these chitinases are used in defense processes has different targeting. There are targeted to the vacuoles, and when the tissue is damaged, these vacuoles, the chitinases from these vacuoles are used for the degrading of the present, for example, fungi.
All right. I think that's everything. I think we got all the questions. Victoria says thank you. And David says very informative. And thank you, Jana, for presenting for the ICPS. And thank you, everyone, for attending this live. And this will be posted on YouTube very shortly so everyone can see.
Thank you. It was a pleasure for me too.
It's not a surprise that gardeners, educators, and scientists are fascinated by these unique plants. The International Carnivorous Plant Society, or ICPS, not only love these plants, but welcomes growers just getting started all the way through professional scientists. The ICPS even started an annual World Carnivorous Plant Day to celebrate them. The free online event is held the first Wednesday of May. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.